Welcome to the PCR and Covid-19 testing presentation. PCR stands for Polymerase Chain Reaction. In this presentation, we're going to do a DNA review. DNA is related to the polymerase chain reaction because the chain that we're growing is a strand of DNA. We're going to discuss how Taq polymerase enzyme is used to build that DNA, and then we're going to talk about how PCR is utilized within RNA applications such as Covid-19 testing. So first, let's discuss the structure and naming of DNA units. So a DNA unit is referred to as a nucleotide. So this nucleotide has a phosphate. So we have a monophosphate, one phosphate group. And we have our sugar, our deoxyribose sugar. It's deoxy because we don't have an alcohol functional group on carbon number 2 of our ribose sugar. And we have adenosine. So adenosine is the nitrogenous base attached to the sugar, or adenine is just the nitrogenous base. So for the nitrogenous base, for adenine, if we add a sugar on here, we'll refer to that as deoxyadenosine. And then if we add the phosphate, we have deoxyadenosine monophosphate. For nucleotides, that means we have our phosphate group, our sugar, and our nitrogenous base. If we have a nucleoside, we have our nitrogenous base and our sugar. And for our nitrogenous base, that is simply just the nitrogenous base. So let's look at the other DNA units that we have. The next one is deoxythymidine monophosphate. So notice that our phosphate group is the same, our deoxyribose sugar is the same, but our nitrogenous base is significantly different. Here we have 2 rings, here we have 1 ring. Our functional groups are a little bit different as well. So for the next nitrogenous base, we have deoxycytidine monophosphate. And for the next, we have deoxyguanosine monophosphate. When we form our DNA strand, we're actually combining a monophosphate unit with a triphosphate unit, as well as an enzyme to facilitate this reaction. We'll discuss the enzyme in a little bit more detail later, but first, let's just look at the arrow pushing mechanism to form our extended DNA strand. So for our adenosine monophosphate, so our adenine nitrogenous base is here and our adenine nitrogenous base is here. So it looks like what we're doing is converting this alcohol functional group to something else. Or essentially we're attaching a phosphate to that alcohol functional group. We also have a byproduct here of our PPI, or inorganic, hence the I right here, pyrophosphate, hence the PP. So 2 phosphate groups, our inorganic pyrophosphate is a leaving group. So for this, we're definitely forming a bond between the oxygen and the phosphate here, because essentially all the rest of this structure here is the same, and that is what we're adding right here. So for the arrow pushing mechanism, we would draw an arrow from the oxygen to the phosphorus. These electrons would come up, and in the next step they'd come right back down, and we would kick off this oxygen or break this oxygen-phosphorus bond. So these electrons would go here to the oxygen, hence how we have our oxygen with a negative charge here. And most importantly, we formed a bond between this oxygen here and this phosphate. So that is this oxygen here and this phosphate here. As the DNA strand grows, it always grows at the 3' position. So the alcohol functional group is what is always doing that nucleophilic attack on our phosphorus of our triphosphate nucleotide derivative. So for DNA strand representation, we're not going to draw out all these molecules every time, just because when we look at the PCR reaction, it'd be a little bit overwhelming to see that many molecules. So we're going to represent these with essentially a color-coded block strand. So in this case, we have our adenine, thymine, cytosine, and guanine in a sequence going from the 5' to 3' direction. So my representation here is going to be a block where this long rectangle is my deoxyribose sugar and phosphate backbone. My adenine is going to be represented by the ash color of this other small rectangular block. Thymine is going to be turquoise, cytosine is going to be caramel, and guanine is going to be green. I chose these colors so they'd match up with the first letter of our nitrogenous base, and for artistic purposes, of course. All right, for DNA base pairing, this is the next step. So DNA strands are usually double-stranded DNA. So usually our strand is base paired with another strand. So again, the 5' is the numbered carbon on our ribose sugar. So we have carbon number 1, 2, 3, 4, 5. On carbon number 5, we have our phosphate group attached. And the 3' position is where the alcohol is linked to a phosphate, or we just have the alcohol functional group. So here we have carbon number 1, 2, 3, that's attached to the oxygen. In this case, it's attached to a phosphate. In this case, and that case as well, it's also attached to a phosphate. Here, the 3' position is attached to the alcohol. That is the 3' end, and it will always be attached to an alcohol at the 3' end. Now, I've actually drawn another DNA structure and flipped it so the 3' end actually starts at the top and the 5' end is at the bottom, which is necessary so that we can actually facilitate some hydrogen bonding between our DNA-based units. So here on this we have adenine, where we have our NH2 and our nitrogen, and here this nitrogenous base is thymine, where we have our oxygen and our NH. So that hydrogen can hydrogen bond with the nitrogen here, and this hydrogen on our NH2 can hydrogen bond with the amide functional group, or specifically the oxygen of the amide functional group here. So on the next slide, let's look at exactly how that looks once we facilitate those hydrogen bonding interactions. So here I'm showing the hydrogen bonding between the hydrogen of the NH2 and the oxygen of the ketone, or the amide functional group there. And here is the NH, specifically the hydrogen bonding to the nitrogen here. The next set is our thymine basically got flipped, and our adenine is now on this side, and the same hydrogen bonding interactions happening there. For the next one, we have our cytosine here and our guanine here. We have 3 hydrogen bonding interactions: hydrogen, hydrogen bonded to an oxygen, nitrogen, hydrogen bonded to a hydrogen, and oxygen, hydrogen bonded to a hydrogen. And that's repeated down here where my cytosine and guanine are simply flipped. So the way we're going to represent this interaction is our blocks are going to be pretty close together, showing that there's hydrogen bonding interactions. For all of my AT interactions, we have 2 hydrogen bonding interactions, and for all my GC interactions, we have 3 hydrogen bonding interactions. So it's important to note that our bonds, our DNA duplex is a little bit stronger the higher our ratio of GC hydrogen bonding interactions we have. So essentially, the more cytosine and guanine you have, the tighter or more strongly your DNA duplex will adhere to itself. So now let's look at this and think about an introduction to PCR. So for the PCR reaction, we're essentially building DNA. So we're building more DNA strands. When our DNA duplexes are hydrogen bonded to each other, we can't do that. So we need to first denature the strands, specifically separate those strands and expose the nitrogenous bases so that we can build more from our triphosphate starting materials that we would have. So right now I'm showing the hydrogen bonding interaction in our strand tightly bound together. We need to denature those strands by heating our vials up to 94 degrees Celsius to separate the strands, which are shown over here. Once we separate those strands, basically what we're doing is eliminating the hydrogen bonding interactions. When we heat it up to 94 degrees Celsius, that's enough to do that. Free rotation of these bonds and the movement that the 94 degrees Celsius provokes within that molecule is going to no longer allow the hydrogen bonding interaction to occur. So we would call that a denatured strand, where the hydrogen bonding interaction is no longer happening. That's the first step of the PCR reaction, is denaturing the strands. In the next step, we need to start the chain reaction. And to start the chain reaction, first we have to have primers bind to the complementary sequences of the DNA template. So we've got our DNA templates now. So our single-stranded DNA is here and ready for the primers to bind. So when we look at these primers, we need to match these primers up with somewhere on the strand that will hydrogen bond with these primers. We've dropped the temperature now down to 55 degrees Celsius. So if we look at where this would hydrogen bond to, it looks like it matches up pretty well with about right there. And for this one, it looks like there's nowhere else on this strand that would have the hydrogen bonding interactions we're looking for. We're close over here, but it looks like our colors don't quite match up. So let's flip this around and see if it matches up anywhere over here. There we go. So now we've got our adenine matching up with our thymine, and we have our guanine interacting with our cytosine, and we've got two adenines, two thymines here, two guanines here, and two cytosines here. At this point, our primers have annealed to the DNA strands that we had separated. So the primers have become bound or annealed, or they now have a hydrogen bonding interaction with our strands that we want to duplicate. Once we have those primers on, that gives a foothold for our DNA polymerase, our Taq polymerase, to interact with the DNA strand and continue to grow that DNA strand from the 3' end. So imagine we have an alcohol functional group on our thymine here. That alcohol functional group on the thymine, on the ribose sugar, is going to attack the phosphorus here. We're eventually going to break this bond right here and kick off our pyrophosphate. So our inorganic pyrophosphate, or PPI, is going to be kicked off. And we're going to add our guanosine here. So essentially, we're going to build another block here and add a green block to our strand. And that process is going to continue as we build this all the way to the 3' end of what we're building, or the 5' end of our template. That's going to also happen up here. So in the next step here, we're basically going to do the same thing, where we're also going to add our guanosine triphosphate. We're going to kick off 2 phosphates and end up adding our guanosine monophosphate here, and this strand will continue growing all the way to the end, which will be the 3' end of what's growing, or the 5' end of the template. Now if you look closely in here, we've got our Taq polymerase. We've got our alpha helix structures, we've got some bladed pleated sheets. Essentially we have a protein structure of our Taq polymerase. This green duplex here is a DNA duplex, which has separated, building our DNA base pairs from the primer, or from the growing DNA strand, one by one. So this is the enzyme that is actually building or performing that polymerase chain reaction to extend our DNA strand. So what we get is 2 double-stranded DNAs, and then this process continues to go to the next step. So we're going to separate our double-stranded DNAs to make single-stranded DNAs. Our primers are going to anneal to that once we drop the temperature back down. Once those primers anneal, our Taq polymerase is going to grow those DNA strands, and we're going to make another set of DNA strands. So if we start with 1 double-stranded DNA, we're going to make 2 double-stranded DNAs, which I have shown here. If this goes around the cycle one more time, in the next step, we're actually going to make 4 double-stranded DNAs. So this is going to double every single time. So another way to mathematically look at that is if we start with 2 to the 0 power of DNA strands, which is 1 DNA strand, we'll make 2 to the 1st power of DNA strands, which is 2. So this is the process that I've shown as making 2. In the next step, we would make 2 to the second power, which is 4, and that number would keep increasing by 1 integer every single time. So we'd have 2 to the third, then 2 to the fourth. If we do 30 cycles of this, it would take approximately 2.5 minutes per cycle. If we did 30 cycles of this, we would end up with over 1 billion double-stranded DNAs. So please punch that into your calculator just to double check. But if we did 30 cycles, starting out with just simply 1 double-stranded DNA, we would end up with over 1 billion DNA strands. So that is what's so amazing about this. We can essentially take a small amount of DNA and amplify that to have over 1 billion DNA duplexes running through this Taq polymerase cycle. And like I said, it takes only about 2.5 minutes. So if we do that 30 times, it would only take about 1.5 hours to amplify our DNA by that amount. So let's take a step back and look at this in lab. So in lab, this all seems a little bit overwhelming, but it's actually very simple as long as you have all the right starting materials. So we need our triphosphates. We definitely need our Taq polymerase enzyme, which is definitely sensitive, so we have to store that in the freezer. We don't have it out for very long. We don't do any thorough mixing. We essentially add about 1 microliter of our polymerase mixture into our vial over here. Once we add in everything else that we need our DNA primers and we need our DNA sample. And that's pretty much it, except for our PCR reaction buffer, which includes some buffer salts, magnesium salts, and potassium salts to allow our DNA to interact appropriately with our Taq polymerase enzyme so we can build those DNA strands, keep everything relatively stable, and allow that multiplication to occur. Hopefully you notice that we do these steps at different temperatures. So to quickly adjust the temperatures from one to another, so to start out at 94 degrees Celsius, quickly drop it down to 55, let that hold there for as long as that needs, 30 seconds to a minute, and then increase it to 72, hold that for as long as it needs. We need a thermocycler. So these are two examples of thermocyclers that we have in the lab that quickly do those temperature adjustments. So each of these reactions is optimized as it cycles through each step in our PCR reaction. So in conclusion, we essentially mix our starting materials into our vial, put the vial into our thermocycler, and pick the right program to run it, and it allows that reaction to form over 1 billion DNA duplexes. So that is the summary of the PCR reaction. Now, like I said at the beginning, I was going to relate this to COVID-19 testing. So for COVID-19 testing, what we have is a virus, and within that virus, it actually does not have any DNA in it. The COVID-19 virus only contains RNA. For testing for the COVID-19 virus, the first thing that we need to do is isolate RNA from the actual virus by lysing the cell and then performing membrane filtration so that we only get our RNA that we're looking for from the sample. Once we've isolated that RNA, we use reverse transcriptase to build complementary DNA strands. Reverse transcriptase is an enzyme very similar to the Taq polymerase enzyme, except that for the reverse transcriptase enzyme, it prefers to build complementary DNA from an RNA template. Looking at the RNA structure here, the RNA is a little bit different than the DNA, so we've got the red backbone for the RNA. Specifically, the differences are that we have an alcohol functional group on carbon number 2 of our ribose sugar, so it's no longer a deoxyribose sugar, it's a ribose sugar. Also, for RNA, instead of using thymine, like with the DNA, we actually have a modification where we have uracil. So this is the nitrogenous base uracil, which does not have the methyl group right here that our DNA has. So if this RNA structure was going to form a hydrogen bonding interaction with a DNA structure here, our uracil would still hydrogen bond with adenine because that methyl group would have minimal, if any, difference. And here is a quick review of what our thymine looked like. So that is thymine with that methyl group here. So here we have our DNA strand. Our thymine is that blue color. That's still going to interact with adenine. For our RNA, we have uracil instead of our thymine, and that will still hydrogen bond with adenine from our DNA. So for our reverse transcriptase enzyme, this process is somewhat similar, where we're going to add our triphosphates, and we're going to start with a primer and continue building our complementary DNA strand from that primer from the 3' position. So in the next step here, if we're going to build on the 3' end, we're going to take the alcohol functional group of our cytosine, and we're going to add our guanosine triphosphate, kick off our 2 phosphate groups, and have our guanine as the next base for the growing or complementary DNA strand. Once we have that complementary DNA strand, once we've formed our duplex, we need to denature that strand or separate that strand out so that we can jump back into the PCR cycle and anneal our primers to selected complementary sequences of the DNA template. Now, this may take a little bit of research for you on the side to get a better idea, but for the COVID-19 virus, there are actually 30,000 base pairs in the RNA strand of that. So for our primers, we need to be very selective and make sure that our primers are only going to interact with the area or the specific base pair region of RNA or complementary DNA that we want to focus on that is specific to our COVID-19 virus that we want to test for. So oftentimes our primers are many more than just 6 base pairs. They could be 8 or as many as 16, depending on how specific we want to make sure we are for a specific region of a virus that we want to duplicate. Also, we've added an attachment probe. So let's look at this. The primers are going to bind in the same way that we talked about before. So let's take this one down, make sure that matches up. It looks like it does. So our primer would bind here, and this primer would bind in this case to our RNA. My mouse is a little jumpy there, but it looks like we've got good base pairing happening here. So that is going to bind there. But our attachment probe is also going to bind. So the attachment probe contains a fluorophore dye marked with the F and a quencher marked with the Q. Let's see if this can perform some hydrogen bonding somewhere here. It looks like it can right here. So our guanosine and cytosine are going to hydrogen bond, and our thymine and adenine are going to hydrogen bond with each other. When this attachment probe is on here, that fluorophore would often emit fluorescent radiation from it, but the quencher is going to absorb all of that fluorescence that it emits. So at this point in the process, no fluorescence is being emitted. What we need to do is build our DNA strand, break this off so that our quencher is no longer in proximity to our fluorophore, and this will make a little bit more sense in the next step. So we're going to have Taq polymerase in our mixture so we can grow the DNA strands. What it's going to do is overwrite our attachment probe. So this is going to be broken up because our Taq polymerase is going to prefer to adhere to our primer and continue growing this strand all the way across. So as it grows this strand, this attachment probe is going to dissociate one nucleotide at a time. So we're going to break off our guanine, we're going to break off our cytosine. Our quencher is no longer going to be in close proximity to our fluorophore. We're going to grow our duplexes, our DNA duplexes. We're going to grow DNA here. This is going to keep cycling back through, but as we keep amplifying this, our number of DNA strands will continue to grow, where our RNA obviously will not grow. And most importantly for this test, it's not that we want to really amplify the DNA of it. What we want to do is make sure that we've released all this fluorophore. So we're amplifying the DNA, and in that process, all of the attachment probe starting material that we have in solution is going to bind to the strand that we grew, so the complementary DNA strand from the RNA sequence that we expected to be in there. But as it grows, we're going to break apart this DNA chain. So our single-stranded DNA is going to break apart, which means that our fluorophore is going to be alone in solution. So it's going to be free-floating in solution, no longer tightly bound to the quencher. What that means is that there's going to be a fluorescent signal emitted, and we can analyze that by a fluorimeter or a fluorescence spectrometer, or better yet, within real-time PCR or qPCR, we can detect the fluorescence emission by hooking this up to a computer, having the right program, and we can watch the fluorescence unit grow as the number of PCR cycles continues. So if originally we did have the RNA strand that was the COVID-19 virus, which means that our Tachman probe here, our Tachman probe has something to bind to so that it can be overwritten and broken apart in the process, then we will continue releasing that fluorescence signal, and the fluorescence unit will continue to grow in our analysis program that's attached to our qPCR device. So that is exactly how our RT-PCR testing for COVID-19 works that's performed all over the nation right now. We are detecting the fluorescence signal emitted, so really all we have to do is put our sample in the vial, add in our Tachman probe, our triphosphate units, and our Tach polymerase, as well as our buffer, allow this to go, and the reading of the fluorescence unit, if it expands, there are some additional calculations related to this, but it's pretty much this simple. If the fluorescent unit continues to grow with the number of PCR cycles, so essentially if we see a spike here, then likely there was the RNA of COVID-19 in the sample. If it does not, if it flatlines across or there's only slight growth because over time probably some of the Tachman probe could theoretically be disrupted, then that means that there is not a positive test for COVID-19.